Thank you so much. I'm really excited to be here today. Can you all hear me? Okay, so I'm going to share screen, presentation mode. Okay. And my email address should be in the chat. I am happy to answer any questions you may have. Before we get started, I have no relevant financial relationships to disclose. And I would also like to acknowledge my colleagues and team of co-authors, Dr. Young Bae, my advisor and expert in cleft palate research, Dr. Eileen Kalmar, an anatomist and my committee member who has personally dissected over 2,000 heads. And I think most of us know Carrie Obert. And I would also like to thank the Body Donor Program at The Ohio State University for providing me the opportunity to spend a full year in the cadaver lab. So I will be presenting our 3 publications today, and the parts of those involve either cadaveric or MRI research. So I want to talk a little bit about my cadaveric dissection experience first. I think everybody present here today has some interest in anatomy, but we're used to these very colorful, organized, well-labeled drawings in our textbooks. And then when you actually get in there, you realize it's going to be a much steeper learning curve than you thought. So the reason I set foot in the cadaver lab in the first place was to get that experience to make me better at reading MRI images. And we're going to look at several of those today too. The plan was to spend 1 semester, but that quickly turned into a full year of dissection. So you will see some images. I have hundreds and hundreds of pictures, and you will see just a few of my images today. But before we look at the cadavers, I do want to go through a brief anatomy review. And Netter's is the anatomy resource used in the anatomy department at Ohio State, and it is the one that I have come to prefer. So we're going to look at several Netter's plates here first. So here is our velopharyngeal mechanism. This is the right side. Okay, these are teeth. Here's anterior, posterior, superior, inferior. You can see my cursor, correct? Okay. All right, so I'd like to point out, so here we have the tendinous portion of tensor and the muscle fibers here. Tensor, of course, tenses the velum. It makes your ears pop because of its attachment to the membranous eustachian tube. We have levator coming in here and inserting into the velum at its oblique angle. That's going to be very important for one of the images, or for one of the studies I'll be showing you. Here we have the cartilaginous eustachian tube, which is also known as the torus tubarius. And the cartilaginous eustachian tube is the origin of the salpingopharyngeus muscle, which we will be talking about at length today. So tensor tenses the velum. Levator is our primary muscle of velar elevation, and salpingopharyngeus, well, we will get into that. Now here is our first cadaveric image. So this is the same view, except now you're looking at the left side. So anterior, posterior, velum, hard palate. The image on the left is the intact mechanism after the head has been bisected down the middle and opened like this, and the overlying mucosa here is still intact. We see a prominent salpingopharyngeal fold. This indentation here is the opening to the eustachian tube. You can insert a probe in there and appreciate its depth that way. The image on the right is what we have after that overlying mucosa has been very carefully removed. And underneath that, we see a very large salpingopharyngeus muscle. Here we have levator coming in at its oblique angle, and we can appreciate the tendinous portion of tensor here. And we could have continued the dissection to see more of tensor. Tensor is still underneath all of this. So moving on, in this region, importantly, we also have superior pharyngeal constrictor. And I think we know our pharyngeal constrictors, we have superior, middle, and inferior, and those muscles essentially overlap to create a very large and long sheet of muscle. But one thing when you dissect, you really begin to realize is one of the limitations of studying anatomy out of a textbook is you're translating these highly complex 3D structures to a 2D image. So to appreciate your pharyngeal constrictor here, the whole thing is a C shape. It would be coming out of the screen at you. This plate has cut. Here's a cross section of the fibers of the superior pharyngeal constrictor fibers here. And I'd also like to point out your pharyngeal constrictors get progressively thicker. So as you move from superior to inferior, by the time you're at the inferior pharyngeal constrictor, that thickness is actually quite massive. Another smaller point of interest here is the palate-pharyngeal ridge, also called the palate-pharyngeal sphincter or sometimes passivance ridge. That is a raised band of muscle fibers moving in that C shape along the medial surface of superior pharyngeal constrictor. And there has been some recent work on this, and it is thought to assist with the cinching action to close the VP mechanism. So when we talk about VP closure and having sound not come out our nose, we spend a lot of time thinking about raising our velum. But if we're thinking in 3D, the cinching action of the superior pharynx around the velum to fully close the VP port can be just as important. And that's part of what that little ridge does. When we look at our MRI images, you will appreciate the levator sling forms a V shape as it comes in on both sides. And then mirroring that, we have palatopharyngeus going from the velum inferiorly into the pharynx, and the palatopharyngeus forms an upside down U shape in the throat. And the palatopharyngeus helps form our longitudinal pharyngeal muscles. So in the literature, the pharynx has been described as a muscular hydrostat. And what that means is you have many groups of muscles with many muscle fibers that run in different directions. So your superior, middle, and inferior pharyngeal constrictors, the fibers run this way. But then you have your longitudinal fibers running this way, and they're made up of three main actors. So palatopharyngeus, your upside down U shape. Salpingopharyngeus, which I showed you, salpingopharyngeus interdigitates and connects with palatopharyngeus. And then from the outside of the pharynx, we have stylopharyngeus, which originates at the styloid process at the base of the skull. And that one is the only muscle that originates outside the pharynx and courses inward. So those three muscles come together kind of like prongs on a fork to create our longitudinal pharyngeal muscles. Another point of interest here, we see our aryepiglottic muscle fibers coming around the arytenoids here. So this is a posterior coronal cross section of the pharynx. And if you look at the larynx here, this is one of my cadaver images. This is the same view I just showed you. It's just rotated slightly. So we have cricoid cartilage. Here's your posterior cricoarytenoids, transverse interarytenoids, oblique interarytenoids. Then we see some aryepiglottic muscle coming off of the arytenoids here. And in most cases, it is accurate to think of the aryepiglottic muscle as a continuation of those oblique interarytenoid muscle fibers. But what we're seeing with aryepiglottic muscle is that, and really most of these muscles in general, is that do we all have a beautiful voice? Yes. Do we all have the same parts? Yes. But there can be significant individual variation in exactly how we're put together. And it's really important for us to understand the anatomy, the variations in the anatomy, so we can have a thorough understanding of our physiology. This is the same specimen in a different view. This is your superior view, similar to your endoscopic view. Here I am holding the epiglottis here. Here's your pharyngeal constrictors, your sheet of muscle here, but I have bisected it with scissors. But what I want to point out is this coming off the arytenoid here, here's your aryepiglottic muscle. But what we see with the aryepiglottic muscle is its course continues quite laterally. And in this particular instance, it becomes continuous with those longitudinal pharyngeal muscle fibers. It does not curve around and insert directly into the epiglottis. Let me show you another publication that looked at this. So this is a fairly recent study from the swallowing literature, and these images detail some individual variation quite beautifully here. So case A, here's your bisected cricoid cartilage. Your blue arrows are your aryepiglottic muscle fiber. Red arrows is your longitudinal pharyngeal muscles. And at this point, it's a combination of stylopharyngis, palatopharyngis. You would actually have to do staining to know exactly where those muscle fibers came from. But in case A, the aryepiglottic muscle fibers are pretty sparse. They terminate about here and they don't actually interdigitate with the pharyngeal muscle fibers. And you'll also notice we see our aryepiglottic fold along here, along here, along here in these three cases. And our aryepiglottic muscle fiber is inferior and a bit lateral to the actual aryepiglottic fold. So in this aryepiglottic fold, there's no muscle fiber in this part. The muscle fibers are out here. Case B, we have a quite large and prominent aryepiglottic muscle that is clearly interdigitating with our longitudinal pharyngeal muscles here. And then case C is a bit of a hybrid. The lateral portion connects with the the longitudinal pharyngeal muscles, and the medial portion is shorter and terminates about here. So I can confirm from my experience that this is pretty typical of the individual variation you might see. The one that I showed you was more like this, but one of my other specimens looked more like case A. So now we're at our first of the three papers we're going to be discussing. This paper was not sent out in the email ahead of time because we just learned last week that we've been accepted to JSLHR, and we're very excited about that. And I wanted to start with this one because it's the newest and it's the most recent, but I want to use it to also give you an overview of here's what cadaveric and MRI research involves. Here's what that process looks like. So, I'm in my first semester of dissection, and my manual tells me I'm gonna start dissecting the soft palate, the velopharyngeal area for the very first time. And the dissection manual says, step 1, identify the salpingopharyngeal fold. And I thought to myself, as a speech pathology student, I remember reading in my textbook that the salpingopharyngis is not always present. And even if you do have one, it probably doesn't do anything important anyway. So I asked Dr. Kalmar, does my specimen have a salpingopharyngeal fold? And she looked at me like, yes, of course. Why would it not? And then we quickly discovered that we have a stark discrepancy between the speech science literature and the anatomic literature. The anatomic literature does not acknowledge the possibility of the salpingopharyngeal muscle being absent, and it talks about it as part of that trio of longitudinal pharyngeal muscles that elevate and widen the pharynx during swallowing. So quickly we realized that we needed to do something about this. And in reviewing the literature and digging, our speech science texts tend to cite the same landmark 1972 speech science study that stated the salpingopharyngis is not always there and not important. But through our work, I think we're able to kind of explain and rectify some of this. So some variables to consider that throughout the literature are inconsistently reported, but that do matter, are number one, fix. So the way the cadaveric material is preserved, you have three basic options. You have the traditional fix, which contains formaldehyde. That is inexpensive, it lasts a long time, but it dehydrates the tissues and makes them very difficult to separate. At Ohio State, we have a proprietary solution called the anatomical fix. That one does not last as long, but it maintains some flexibility in the tissue. So on your anatomical fixes, the fingers can still move, but on the traditional fixes, they don't. Then, of course, you have fresh tissue, which I have not worked with yet, and that does pose some logistic challenges. At Ohio State, we also have clearly identified characteristics. So we know the biological sex of our specimen, we know the age and the body weight at time of death, and we know a basic cause of death. And then the dissection approach refers to step by step, how did you access that particular area or that particular muscle? Okay, so my cadaveric sample. I started out with 24 cadavers. I could only use salpingopharyngeal muscles from 19 of those, and of those 38, I could only use 31 of them. Is it because they weren't there or originally present and intact? No, it is because they were shared with first-year medical and dentistry students who were, most of them, fresh out of undergrad and embarking on their first dissection experience. And this is what you look like after you have completed your very first semester of dissection. So, there were scalpel cuts in places where you shouldn't use a scalpel, etc. But what we had our information, our sex information, age, body weight, and our fix information. We had all of that. And then to access the salpingopharyngeal fold, we use the same step-by-step approach for every single one. So step one is to bisect the head. That is done by hand with a large saw. The medical and dentistry students did do that part for me. I did just about everything else. But it is important in that initial saw cut that be perfectly center, because there was an instance of an off-center head bisection where the saw cut went clean through the salpingopharyngeal fold and we lost the contents that way. Then we took digital calipers and we took width measures across the salpingopharyngeal fold before and after removing the mucosa. So I will show you exactly what this looks like. So here's a similar image to what we saw earlier, tongue, velum. Here's your salpingopharyngeal fold. The overlying mucosa is intact. Here is your digital caliper. And what you can do is take a gloved finger and palpate along the length of the fold. You can feel the cartilage at the origin point up here, and then you can feel where you have just muscle base a little bit further down. And we took width measures across both points. In this picture, you can appreciate just how thin and delicate the mucosa is. So we see the mucosa stretched over the scissors here. And especially in your traditional fix, it's very difficult to peel that mucosa off without also pulling up muscle fiber, which you really don't want to do. So once your mucosa has been carefully removed, here we see our cartilaginous eustachian tube, or our torus tubarius, and the salpingopharyngeal muscle running into the pharynx. Here is its anterior interdigitation point with palatopharyngeus. Here is its posterior interdigitation point with superior pharyngeal constrictor. We were not able to come up with a uniform definition for salpingopharyngeal length, but I can tell you, because of individual variation, most of the time, the interdigitation with the pharyngeal wall is at the level of the superior constrictor, but in some cases it's quite long, even at the middle or even the inferior pharyngeal constrictor. Okay, so after looking at our cadaveric variables, we ran a regression and we found that sex and the relationship between age and body weight of the cadavers are significant predictors for salpingopharyngeal muscle size. The same was not true when measuring across the cartilage, which makes sense because cartilage would not respond to age and disuse-related atrophy in the same way that muscle fiber would. So then obviously we have some missing data in our cadaveric sample. So we replicated what we did in vivo using MRI images. And this is the first in vivo report of the salpingopharyngeus muscle. And these were 3D MRI scans of data we used retrospectively. We already had them. So these were not singers and they were not vocalizing at the time of scanning. They were just laying there. And now we have our first MRI image. So let me orient you to this process a little bit. So in our lab, we use a software program called Amira. And what Amira allows you to do is you take all your stacks of images and you reconstruct them in a 3D space, and then you slice them, and then you can select pixels, you can draw lines on them and make your measurements. These, what you're looking at are T2 weighted images. So black is airspace, cartilage and bone are white, and muscle fiber and other things are the shades of gray in between. And so when you're slicing MRI data and selecting exactly what image you need to analyze, you have to have anatomic reference points for what you're doing, because the way the person was positioned in the scanner can make a big difference. So slice A and slice B are these axial slices, these cuts here, but the slice has been pulled out and rotated. So you see this oblique axial slice. And for this, we defined that as the hard palate line cut and lifted out. And then here at the level of the cartilage or the torus tubarius, you can see a white cartilage that is the origin of the salpingopharyngeus muscle. We took our width across there. And then the inferior slice, the lower one here, you see a clear gray muscle bundle. And there is your salpingopharyngeus here. And then you can also appreciate levator and tensor in there as well. Took our width across here. So the way we took our width measures mirrored what we did with the cadavers. Here is a parasagittal MRI image that is identical to the cadaver image I just showed you. So in looking at this, it does, especially at first, just looks like a bunch of gray blobs. So you can appreciate how some cadaveric dissection experience would help train your eye at being able to read these. So here's the tongue, here's the velum. 5 is actually your levator bundle inserting into the velum. 4 is palatopharyngeus running down here. 2 is salpingopharyngeus, and then the green arrow shows you where salpingopharyngeus interdigitates with palatopharyngeus. 3 is our superior pharyngeal constrictor coming down here, and then the red arrow details where salpingopharyngeus interdigitates with pharyngeal constrictor, in both cases at the level of superior pharyngeal constrictor. And these are the same, these are just two different subjects. So in what we did, our approach was replicated between the cadavers and the MRI sample. So the numbers were pretty close. So your cartilaginous width is somewhere 5.5 to 6. The width of the salpingopharyngeal muscle itself, about 3.5 to 4. Compare that with the muscle we're looking at next, levator. Levator is about 5 millimeters wide, and levator has received a good 20 years of MRI imaging and research attention. So, but the important point I want to make here is that the salpingopharyngeal fold and muscle were identified bilaterally in all 15 subjects. So yes, you do have one on both sides. And I think our paper serves to explain what happened and rectify some of the discrepancy, as well as also appreciate the significant individual variability that we have. And again, it is really important to have all of our anatomical facts ironed out before we can really have a good understanding of physiology. So this is our CPCJ paper. These, what I'm going to show you are the first images, MRI images of the contracted levator sling in vivo. And they will actually be featured on the cover of the journal when the issue is released. So we're very excited about that. So how did we get the first images of the contracted levator sling? Well, that was thanks to our 4 lovely Astilians, all EMT or higher, and they were asked to do a gargantuan task. The scan was 4 minutes long, so they had to hold their velum in the elevated position while breathing out of their mouth, in and out of their mouth, in a silent E gesture for 4 minutes while laying on your back and a noisy MRI. So they were given this assignment a couple weeks ahead of time. They practiced in the mirror, they laid on the floor to silent practice, hold their velum up while saying their silent E. And it did work. So this figure, if you had a chance to review the papers before the meeting today, this figure is not the final figure that was published. I will tell you why in a second. But these are the 4 participants that we had. Okay, so here, the rows 1 and 3, here's your mid-sagittal image. These columns are the velum at rest. You see the velum laying on the tongue base here. And then these columns are the velum for silent E. So you can see the velum clearly elevated and contacting the posterior pharyngeal wall. And oops, you can clearly see that in all 4 of our participants. Now these rows, your oblique coronal image, that is probably looks a little bit more foreign. So let me explain what you're looking at. I showed you how levator comes in at its oblique angle. So the oblique coronal slice is basically like this. So it goes straight, it follows the course of the levator muscle going straight through it. And then this slice has been lifted out. So what we're looking at here, you can see this dark V shape. This is the levator from both sides coming in and inserting into the velum. At rest, levator is in a pointy V shape. Then when it contracts to lift the velum, that V shape goes to more of a U configuration, like this. And that's what you can appreciate here. So at rest, it looks like a V. During silent E, it looks more like a U. So why did we discard a participant? So the journal wanted a short, straightforward article of here is how to get images, MRI images of the contracted levator. Participant 3 did great with the silent practice, held their silent E for 4 minutes, brought the velum against the posterior pharyngeal wall, but what happened? There was little to no change in the configuration of levator. Why? We speculate because if you look, this velum is more straight back, whereas these velums are more arched and up and back. So we think participant 3 actually pulled velum straight back and used the transverse fibers of palatopharyngeus to do that. So participant 3 didn't really activate levator. So levator gets all the research attention. Yes, it is the star of the show of velar elevation, but I think this is very interesting to still present because it shows you how levator does not work alone, and it allows you to appreciate the nuanced movement that you can have within the velopharyngeal mechanism, and that's just one example. So, quantitatively, we found, so we know when a muscle contracts, the muscle fibers get shorter and thicker, and that's exactly what our numbers showed. The levator sling got the extravelar portion, the part sticking out of the velum, got 13.5% shorter, and it got 22.1% thicker cross-sectional. CSA means cross-sectional area. This basically means thickness. And then smaller angle is in relation to the cranial base here. So it basically measuring how it went from this to this, our angle got about 10% smaller. Okay. So, and finally onto our twang study. This one you may have heard about already. This was published back in the spring. So again, we used 5 very experienced Astilians, and they had another gargantuan task. They had to hold E on their most comfortable pitch. We let them decide what that was. And they were asked to do productions in estal, oral twang, and speech quality. And so at this point, I would like to talk a little bit about some of the prior MRI studies that are available on specific to voice. So MRI and cleft is kind of its own field, but the MRI studies specific to voicing tasks have typically involved a very small number of subjects, like 2 to 4 is pretty common. Some of them are just one subject. And in previous voice MRI papers, their idea of a validity test is the subjects and the investigators thought everybody did a good job. And sometimes that is practically verbatim what they say. We chose to take the recorded productions of what people actually did in the MRI and send them to 2 estal trained speech pathology experts to rate them and confirm that yes, our subjects did in fact produce twang. And this process, between the 18 seconds and some subjects losing their twang in that noisy MRI environment while laying on their back, those productions were discarded. They were not included in the analysis. So we had 2 final subjects, a male and a female. Our MRI microphone can filter out the MRI banging to some extent, but as you'll hear, it's clearly not perfect. I'll play you. Here is what the female sounded like. Everybody can hear that okay? Okay, I'll play the female one more time. And here's the male subject. Play you the male one more time. Audio quality is not great. Again, this is after the MRI noise has been somewhat successfully filtered out. This is a production of a subject that the expert rater said, no, that was not twang. So this is an example of one that would have been discarded. Not twanging. And actually, my first draft of this paper actually included the subjects that failed the validity test because their numbers didn't change. The pharynx was wide open and giant, but we ended up just okay, twang chorus. Anyway, we ended up just discarding everything that wasn't twang. Okay. So here we are, we have our mid-sagittal MRI image. And another limitation to prior MRI voice studies is that they will measure larynx height and velar height, but even the same author won't use the same measurement approach from paper to paper. So the cleft literature has 20 years of here's exactly how to measure the levator, whereas in voice, we don't really have that, at least not yet. So what we came up with was to use the hard palate line and put a reference line here. And then larynx height was defined as the perpendicular distance from the hyoid bone to this reference line. So then when you're in a mirror and you draw this line, a mirror tells you exactly how long this was in millimeters. So a longer line means a larger number, means a lower larynx. For velum, we use the tip of the uvula, perpendicular distance to the reference line. So again, a larger number, a longer line means a lower velum, and vice versa. And when we replicated this, some prior studies have not included inter and intra-rater reliability measures. We have several members of our lab who are trained in AMIRA. I did all of the measurements twice, and a lab member replicated them, and our inter-rater reliability had an agreement of 0.9 or higher. And that those numbers are always reported in clef research, but not typically reported in voice research, but hopefully they will be going forward. Okay, so from these measures, we found the larynx was higher for twang. I don't think we're surprised at that finding. In velar height, there was no difference because we asked for oral twang. So in both the twang and the speech, the velum was up, the port was closed. So velar height was more or less the same for both tasks. So prior MRI voice studies have been exclusively limited to this mid-sagittal picture, which I'm showing you right here. So your mid-sagittal image is a slice from the middle of the vocal tract. So you take the middle slice and pull it out and look at it. So you're only getting this. So in that, you're potentially missing all of the movement and the adjustment that could be going on out here. So for that reason, and as far as we understand, this is the first time this has been done for voice, we included an axial slice to try to capture some of this activity. So this white line is where we made the slice, and then here it is pulled out and rotated so you can see it. And the definition for this axial slice was the superior mandible perpendicular to the spine, and that line also goes through the tongue base and the tip of the epiglottis. I do want to note that this slice is pretty far above the aryepiglottic fold, and it is above the aryepiglottic muscle. So those are not visible in this image. But what you do see is you see this black vocal tract airspace, and this is a slice of the shape of that throat at this level. So anteriorly, we have the epiglottis and base of tongue, posterior pharyngeal wall, and then medial to lateral, we have the pharyngeal walls here. So here's the corner of the pharyngeal walls, posterior pharyngeal wall, tongue and epiglottis. So we measured the anterior to posterior width and the lateral width here. So this figure shows you the exact 8 images that we used to make our measurements. Participant 1 is male, participant 2 is female. We have our mid-sagittal images, and I think in the twang productions you can appreciate that the larynx is in an elevated position. Then we have our axial slice, which is what you see here. Look at the male subject. So the overall shape of the vocal tract airspace is the black hole, basically, that you see. But in the male's speech production, his black hole is quite large. And this faint gray line you see here is actually the tip of his epiglottis. So that means this part of the black space. So here's the tongue base. Here's the epiglottis. All of this is the space in his vallecula. And then this space is from the epiglottis to the posterior pharyngeal wall. So when making the measurements, you would have drawn the line from here to the tongue base. In the twang production, we see the vallecula pretty much becoming obliterated. These tiny little gray pixels, that might be a little bit of vallecula that's still visible, but we can see there has been a substantial reduction in this anterior to posterior dimension. And we can also appreciate that the corners of the pharyngeal walls have also moved from a lateral to a more medialized position. The female, so I can tell you the female, her speech is already a little bit twangy. You can see a little bit of space in her vallecula during her speech, but not much. But her primary narrowed adjustment was at the pharyngeal wall. So you can see her speech is more of like a oval shape, and in her twang, you see the corners of her pharyngeal walls coming in here. Her anterior to posterior change was pretty negligible, but both subjects had a noticeable amount of narrowing in the lateral to medial dimension from those pharyngeal walls. And again, we're seeing these adjustments at a level that's up above aryepiglottic sphincter. So that's why we propose the term supralaryngeal narrowing, because structures up and around the larynx are also contributing to the reduction in vocal tract area. So the main take-home message here is that, and this has been shown in simulation research also, our perception of twang is related to a shorter vocal tract and a smaller area of the vocal tract airspace, and that is what we're seeing here. So we would, and this is basically what I just told you. So both subjects used the pharyngeal walls. The male subject mainly had some minor adjustments in the tongue base that led to a pretty noticeable narrowing in the AP dimension. And those adjustments combined made the overall area smaller. So, and again, above the aryepiglottic sphincter, we would, and we also have a companion endoscopy study that has been slowed by COVID. And that one has 20 subjects, and that should be out hopefully soon. And so what we're seeing is tongue base and pharyngeal walls contributing to the narrowing of the aryepiglottic area. But as I showed you, those muscle fibers, especially in some people, can be pretty sparse. And in other people, they interdigitate with the pharynx. So what we're saying is the aryepiglottic muscle doesn't work alone to narrow the vocal tract, and possibly it even is not the star of the show for that. So to just summarize the three studies together, so we've learned we have a salpingopharyngeus. Everybody has one. It was a poorly studied and understudied part of the body. Our levator sling, we've been able to see it contracted in living people. And levator, while it is the star of the show of velar elevation, it does not work alone. And I have a feeling that salpingopharyngeus also helps assist in that cinching closing action of the superior pharynx around the velum. And I think that is a pretty good summary. So at this point, I would like to open the floor for your questions.
Karen, thank you so much. And I forgot to thank, yes, woo, woo, woo. We forgot to thank the Ohio State University as well. That's where you're getting your PhD. And I would like to say that your dissections are so beautiful. They are amazing. When Karen was talking about the medical students who cut through everything, that was me in dissection. So I am in awe of how her fine motor skills. She's a wonderful singer. She was always an inquisitive EMT when you were working. I was like, Karen, you have all those questions. It is perfect for you.
It did not start that way. I cried my way through the carotid triangle for the first time. Don't get me wrong. I got a lot. I would like to think that I grew a lot in a year.
I guess so. Like, right, I just quit. Okay, so I had a semester of dissection. I'm like, oh my gosh, that is not my special sauce with my eye-hand coordination. Scalpels, don't give me one. But Karen, they're beautiful. The work is beautiful. It is, again, with your 3 studies. It was nice to tie in the salpingo with the levator and the whole team, right? As we know, there's a team in Estel voice training. Also, it brings home in the courses when we did head and neck control that we would, in the old times before the iPhone, we would run to a big mirror in the bathroom and go, hey, there's a lift, and then there's a lift and a cinch. And
Exactly.
so, yeah, and I'm like, Karen, oh my gosh, the salpingopharyngeus along with palatopharyngeus might be our cinchers. And so, everyone here with your iPhone, have fun with turn on the flashlight and do a lift and then a lift cinch. And it's exciting information, and we're building on models because, as everybody knows, that's all we do have for voice models, right? We're collecting data. These are models to that will evolve. And what questions? I asked my questions to Karen, so I can chime in. But what questions do you have for Karen? This was amazing, and let's go. We have about 10 minutes
Yeah.
for questions. Corinne, are you
Yeah,
collating those?
none came in the private chat, so I'm going to start us off until other people get brave. Karen, what came to mind when the spinal conclusion, you were just talking about, the implications of the narrowing of the pharyngeal wall. We couldn't really see that epiglottis. Are there already studies out there to compare to just high larynx without people attempting twang at the same time?
Without people attempting twang at the same time? That's a good question. Most of the MRI work talks more in like opera, belt, more in qualities, and there haven't been a lot of studies attempting to really isolate. So I'm not aware of a study that looks just at high larynx without twang.
Yeah, I think there's something there. Maybe for future MRI, look at people just performing a high larynx E and then their twang E. And
High larynx to twang E. That would be helpful. And Carrie Obert is the lead on the endoscopy portion of this, which unfortunately has been slowed by COVID, but we are analyzing those results currently, so there will be more to come.
great. Kim McGinnis has a question. Kim McGinnis, she's coming. She's here.
Mic, internal mic. There we go. Okay, I was kind of hand up, hand down. It was more for those of us who are interested in being part of research studies. Do you have any tips for us of how we can prepare or what can make us better subjects?
Oh, that's a really good question. So I think first of all, enthusiasm and curiosity goes a long way, as long as the ability to travel to Columbus, Ohio. These tasks were provided to our participants in advance who all did travel to support us. I think especially the Levator study to an extent supports silent practice, right? Because that's how they held their velum up for 4 minutes. So that's pretty amazing. As far as participating in endoscopy research, not everybody can control their gag reflex. MRI research, you have to be okay with noisy environments and tight spaces, which isn't for everybody. But, I guess having a really high threshold of tolerance for being temporarily uncomfortable would also make you a good subject.
Thanks, Karen. I hope I can come visit you soon.
Yes, thank you. And I think I forgot to mention, so these three publications are essentially the work of my first 2 years. And I'm currently ABD or all but dissertation. And COVID willing, am very slowly collecting my data for that. And that will be on motor learning. So I will have a whole sort of other topic, hopefully, to share with you in the future. I think I see some things happening in
Yeah,
the chat.
Kelly Bird just wrote in the chat, how will this or can this influence our understanding and teaching of AES and twang?
I think it has to do partially with how you could map people of where they may or may not feel the narrowing. So, especially in the case of pharyngeal walls, AES is down lower from where our axial slice was. Our axial slice was here. Some people might feel narrowing, possibly anywhere along this dimension, as well as monitoring for tongue base. So minor adjustments in tongue base might be leading to, I don't want to use the word successful twang, but contributing to making the shape smaller. But anyone who like uses extreme tongue root, like on an MRI, you would see narrowing in the AP dimension, but that's not necessarily a sound that most people would be trying to achieve. Unless, so being cognizant of where the twang or the ring is coming from. Is it slight adjustment in the tongue? Is it the pharyngeal walls along here? And then two, unfortunately, we were on one pitch, but I think those adjustments do differ across the range. And that is something that we'll be looking at more specifically too with some of the endoscopy work.
Great. I didn't see any hands raised or chats. Did I miss anyone's questions? Susan.
Can you see me? Hi. Thank you. My question around the perception of twang when you were doing the MRIs with the female, did you know what kilohertz those particular perceptions were matching?
I wish. So our banging from the MRI made the audio quality too poor to do acoustic analysis, but that is coming, and that is easier to do with endoscopy work because it's not as noisy. So more on that to come. That would have been a great addition that we weren't. That's really challenging in MRI. We weren't quite set up for that. So.
I asked because the male on B flat 3, that would be a little higher in the range than the female on C sharp 4.
Sure.
Regardless
Sure.
of what her larynx was. And I'm asking because I've been in a number of Carrie Overt's workshops where a twang perception on C sharp 4 might be more tongue than piriform sinus.
Yeah, so the female on C sharp 4 would be lower in her range, and there's thinking that tongue might be more active in the low range, whereas she didn't show as much. This female subject on C sharp 4 didn't show as much change in that AP direction, but like I said, I can tell you that subject, her speech is already kind of twangy.
Yeah, all right. Thank you.
Does that answer your question?
It does. I just was hoping that you knew because I was wondering if it was going to be 3,000 or 5,000.
3,000, yeah. Carrie is actively working on that.
Okay, we have a minute or two. Did I miss a hand again? Sanders has a hand, and I think this will be our last question for the evening afternoon.
Karen, thank you for this research. I'm wondering, sometimes we have a prompt where we'll get people to do an apple bite or flare their nostrils, which seems to do things in the face. But I'm just wondering, I perceive that as a way of getting extra cinch at the back. And I'm wondering if you can illuminate any of that, and if that has any relationship to the research you've done.
So I agree with you. Pedagogically, I use that snort, bite the apple, all of those things are, because there's a difference between lift and cinch versus just lift. And we haven't used any of those prompts or those silent practice kinds of things in the MRI, but I do just, I guess, anecdotally feel that those do help with the lift and cinch. And the cinching action, again, is really more about the pharynx than it is about the velum. So you can raise velum, you can pull velum this way, palatopharyngeus, levator is more this dimension. Tensor will tense the velum, but the cinch part really comes from superior pharynx, which probably has something to do with salpingopharyngeus, palatopharyngeal ridge, all of those. So we would need to look at those prompts under MRI or endoscopy or something. I don't know that we're there yet with our imaging to really appreciate adjustments that are that minute. Because one thing I didn't point out, your resolution on the MRI image is about 1 square millimeter. So any adjustments that are smaller than that, you're going to miss them. And there is a limitation to MRI research that the longer your scan time, the better your resolution and the clearer your image, but the longer your scan time. So you're always striking that balance between the shortest scan time with the best image that you can get. So we might need some more advances in technology before really being able to look at that much nuance.
Great. Thank you, Karen, and thank you everyone for your time. Yay!
Thank you.
Or if you're good at the yeah, people are good at the emojis. I'm not. I'm just now. Karen has put her email in the chat that she prefers people to write to. So I'm just copy paste again. So,
And while she's pasting, we are so happy to help her a little bit from the Estill Education Fund, and we have Stephanie Rummel's MRI research coming up too. So we are so again working to fulfill Joe's mission on several levels, and this is just a start. I mean, we're hoping again to build that fund and to be able to again provide some support for research, because as you can imagine, I think everybody's aware of research now with the vaccine, and if there's a choice between finding a vaccine and voice research, I think the funding is going to go to a vaccine, which we need, but we're helping with every little step that we can with the Estill Education Fund. However, we're also helping this entire weekend with the issues involving artists and singers and just humans during the pandemic. So our sales at the Estill store for this weekend, the proceeds are going to benefit internationally, UNICEF Children's Fund and also the Actors Fund. So all sales, we're trying to again help also on all levels, ease some of the burdens of the pandemic as well. And we're happy and trying to contribute as well in that manner. So I am thankful to Karen. I am thankful to Estill and Estill trainers, and this is a start with, I hope, a bright future. And we cannot wait as long as we did from Helen Rosens to have our next research report. I know we will find more and more volunteers to present and more interesting studies. Thank you, Karen Perda. Yay, and Corinne for organizing, and everybody. Did I forget anything, Corinne? I think we're good.
no, go to bed, have a coffee, whatever time of day it is. Thank you so much for your commitment, for showing up. So.
And the symposium's coming up. Will probably be virtual, but think about it. Yes, hint, hint, we're going to extend the abstract submission time. So we have a lot of good abstracts in, but I will be in Pittsburgh, but I'm pretty sure we're going to be mostly virtual, and that's kind of good. I mean, we'll have the whole world can attend, and we can record and share. Thank you very much. Goodbye.